I had explained about structure and function of IgG molecule and subclasses of IgG molecule, as well as I had also explained about the structure and function of IgM molecule. Continuing the discussion on the structure and function of other immunoglobulin molecules, I will take up immunoglobulin A, immunoglobulin D and immunoglobulin E. I will first explain about immunoglobulin A. Immunoglobulin A is found in a dimeric form as shown in the diagram in the serum of any person. The dimeric form is formed because of the joining of two monomers. Just see here, two monomers are joined by the presence of a J chain. And about the J chain, which is called as joining chain, I had already explained about it in IgM molecule. This J chain is a hairpin loop like structure with a disulfide bond within itself, and it has disulfide bond establishment with C alpha 3 or CH3 domain of the each monomer on either side. So the disulfide bond of the J chain with CH3 domain or C alpha 3 domain of each monomer links these two monomers as dimers. As I had mentioned earlier, IgA consists of alpha 1, alpha 2 and alpha 3 domain in the heavy chain, and the light chain as usual consists of variable region of the light chain and constant region of the light chain. And the heavy chains of a monomer are held by a disulfide bond as seen in the diagram. So the dimeric form of the IgA is found in circulation would also get converted into secretory IgA when it is going to make its presence in the secretory fluids. The secretory fluids such as the saliva, nasal fluid, sweat, colostrum. Colostrum is the first milk that is fed to the young one born, and the secretion of the lungs, that is mucus secretion, genitourinary tract as well as GI tract. You know, the lung, genitourinary tract as well as the GI tract are lined by mucus lining, and when the dimeric IgA makes its presence in these secretory fluids like mucus fluid and nasal fluid, etc., it is being protected by an additional chain that is called as secretory chain, which is not found in the serum component of the IgA. Serum component is a simple dimer. Now, as far as the half-life of IgA is concerned in the serum, it is about 6 days, and it is almost about 10 to 15% of the total immunoglobulin in the serum. The dimeric form of IgA will constitute about 10 to 15% of the total immunoglobulin in the serum, and the dimeric form of IgA, which is linked with the J chain, would have a molecular weight of about 600,000, whereas the monomer is just like IgG is about 150,000. The J chain, since it is almost identical to the J chain of the IgM, its molecular weight is about 15,000. It is also a polypeptide chain with the disulfide linkages, and it facilitates the formation of the dimeric IgA molecule. And dimeric IgA molecule is found to be more stable in the serum compared to the monomeric IgA molecule. Now coming to the secretory component. The secretory component is also a polypeptide made up of 5 different domains, and these 5 different domains are compared with the immunoglobulin domains, especially the constant region of the immunoglobulin domain. Therefore, the secretory component of the chain is also considered to be a immunoglobulin superfamily. So here the secretory component which is found is of about 75,000 molecular weight. It is secreted actually in the intestinal lining or epithelial lining of the hepatocytes or intestinal lining in the submucosa area. As shown in the diagram here, you can see the secretory component of the chain is shown with the same color, but it is called as poly-Ig receptor. Poly-Ig receptor. And this poly-Ig receptor is found attached to the wall of the epithelial cell from the inner side towards the submucosa area. Submucosa area is deep within the intestinal lining. It is after the epithelial cell lining. This is the submucosa area, and the submucosa area is rich with the population of B lymphocytes and T lymphocytes. Now the B lymphocytes that are found in the submucosa area are almost about, like anywhere between millions of them, and they secrete per day almost about 2.6 into 10 to the power of 10 IgA molecules per day. That may amount to almost about 5 to 15 grams of secretory IgA in the mucosal region of the intestine. The main reason being the IgA being secreted in such large quantities in this area because GI tract is more prone for the infection by the pathogens, because as I had mentioned earlier, when we consume food or when we drink liquids, the pathogens may enter through our oral passage without our knowledge, and since they continuously invade our digestive system, it needs protection against these pathogens, and hence the IgA molecule is always on the guard in the mucosal lining of our GI tract. Therefore, it is being produced in such large quantities by the plasma cells in the submucosa area. As soon as the IgA molecule is produced, you see here the plasma cell is shown, and as soon as monomer of IgA molecule is produced, there is also the J chain that is produced simultaneously. So the two monomers immediately establish contact with the J chain and become the dimers. So the dimeric IgA molecule starts migrating from the deep area of the submucosa towards the inner lining of the epithelial cells. As it comes towards the inner lining of the epithelial cells with the J chain, the dimeric molecule, it establishes the contact with the poly-Ig receptor. The poly-Ig receptor is the parent molecule of the secretory component of the IgA. And this poly-Ig receptor, with a receptor is attached protruding towards the mucosal area. That submucosa area has large number of these poly-IgE receptors extending out like threads. So the dimeric IgA, on establishing the contact with the poly-IgE receptor, is going to be now engulfed in the form of an endosomal vesicle. So together with dimeric IgA molecule attached to the poly-IgA receptor, it is endocytosed as shown in the diagram. You may see here in the diagram, endocytic vesicle is formed in the epithelial cell, and that endocytic vesicle has enclosed within itself the poly-IgA receptor attached to the dimeric IgA molecule. Now that endocytic vesicle migrates from the submucosal area towards the lumen of the intestine. So lumen is the cavity of the intestine where the epithelial cells are projecting out in the form of villi. So the endocytic vesicle carrying the dimeric IgA molecule attached to the secretory component, that is poly-IgA receptor, comes and attaches to the plasma membrane of the epithelial cell from inside, and it everts out, and by everting out it detaches the secretory component from the poly-IgA receptor, or it cleaves the secretory component from the poly-IgA receptor and releases that together with the dimeric IgA molecule attached to it. Now this IgA molecule attached with secretory component is now copiously secreted into the mucosal lining of the intestine or GI tract. Similarly, the epithelial lining of the nasal passage, the epithelial lining of the hepatic region, the epithelial lining of our tear gland, all these show the similar mechanism of adding the secretory component to the dimeric IgA molecule. What is the necessity of attachment of this secretory component? That we'll see. Now how it is attached, the secretory component? Just look at this diagram. Here you can make out that of the 5 domains of the secretory component, the one of the domain at one end is attached to C alpha 2 of the heavy chain by a disulfide linkage, and the other end domain, that is the terminal domain, in the similar manner is attached to C alpha 2 of the another monomer. This is how the disulfide bond of the secretory chain is established with the C alpha 2 domain of the two monomers, and it runs across the two monomers. By having this secretory chain on the antibody, that is IgA, it is now protected from digestion by the digestive enzymes. The digestive enzymes are prevented from acting on the IgA, thereby the presence of secretory chain has a major role in giving protection to IgA, which is secreted in the secretory fluids. Therefore, it plays a significant role in the digestive tract as and when the pathogens enter in the mucosal lining. Now, most of the time, the IgA helps in trapping the antigens that enter in large numbers in the mucosal lining, and these antigens that are trapped by the secretory IgA are going to be excreted, in fact, through the feces. And another important function of IgA is it is strongly active against the pathogens like the salmonella, Neisseria gonorrhea, Vibrio cholera, and viruses such as polio, influenza, and reovirus. So, the IgA being present there in the secretory fluid readily plays a major role in defending against these invading pathogens. So, it can bind to the multiple epitopes of the antigen simultaneously at the same time, as well as the IgA is approximately, in fact, capable of activating the alternative pathway of the complement, but not the classical complement. And it is observed that IgA has a long lifespan in the mucosal lining of the GI tract. It may even go through enterohepatic circulation. That is, the secretory IgA can get transported from the intestinal lining through the hepatic portal veins to the liver, and from the liver it can come back to the intestinal lining once again through the, the hepatic vein. So this is how it can show repeated enterohepatic circulation and surveillance of the GI tract for the pathogens and react against the pathogens. So therefore, it is very important that by having secretory IgA in the secretory fluids, it not only plays an important role in acquired immunity, it also plays an important role in innate immunity. I had mentioned about this when I had discussed about the innate immunity, because it can bind to the pathogens without a prior notice, and it is a non-specific binding, and therefore it can immediately bring about the removal of the pathogens that enter the GI tract or the nasal passage or the eyes or genitourinary tract or even the lung surface. So it is a very important antibody on the secretory surfaces. So I hope you have understood the attachment of the secretory component in the mucosal lining of the epithelial cells and its importance for the secretory IgA. Now let us move on to the next antibody, that is immunoglobulin D. Immunoglobulin D, similar to IgG and IgA, has only three constant region domains in the heavy chain, C delta 1, C delta 2, C delta 3, with a single disulfide bond across the heavy chain by holding the heavy chain. As it is, IgD molecule is found in a very low quantity in the serum, that is hardly about 0.03 milligram per ml. Therefore, it hardly plays any significant role in encountering the pathogens or neutralizing the antigens. It also has a molecular weight of approximately about 150,000, and its half-life is of about 3 days in the serum. But most of the time it is found attached to the B cells which produce it, and it is found attached to the B cell membrane together with membrane IgM molecule. So M-IgM molecule is the primary receptor of the B lymphocyte. Similarly, together with M-IgM molecule, when I had explained about the co-receptor, I had mentioned about IgD. So membrane-bound IgD together with IgM plays a major role in recognition of antigens that come in contact with the FAB site of M-IgM as well as MIGD. And both these molecules like MIgM as well as MIGD help in activation of the B lymphocyte to in fact produce the antibodies corresponding to the antigen that they have encountered. So therefore, the IgD molecule plays a major role in activation of the B lymphocyte rather than encountering the antigens by being present freely in the serum because of its low concentration. It has about 13% carbohydrate residues, as I had mentioned in the case of IgG and IgM. About 13% carbohydrate residues are found attached to the heavy chain region, that is the C delta 2 domain, making it hygroscopic and water soluble. It is also found that the, the, the B lymphocytes which transform themselves into memory B cells, the B lymphocytes which transform themselves into memory B lymphocyte after they have encountered the antigen and after the activation, and some clone of the population of those B lymphocytes have undergone transformation to become the plasma cells to produce the antibody and develop the acquired immunity against that particular antigen. Now, against the same antigen, that clone of B lymphocyte which had gone activated will become a memory cell. And that memory cell was found to shed off the IgD receptor from its surface. Therefore, some scientists claim that the memory cells shed off the IgD receptors, and therefore it is a necessity for suppression of the B cell activity. The memory cells are suppressed B cells, but they become active only when they encounter the antigen once again if the same antigen enters. So, inactive state of B lymphocyte means which has been now converted into a memory cell has been found to show the absence of IgD receptor on its surface. That is an indication that IgD is not required in the memory B cells. So, it is also observed the antibodies to certain antigens such as insulin, penicillin, milk protein, and diphtheria toxoid and thyroid antigen are found to be of the IgD category but not long lasting. For a short duration, it has been found to be produced and not long lasting. So therefore, primary role of IgD is to be the receptor of the B lymphocyte rather than be in circulation to encounter the antigens. So let us move on to the next antibody, that is IgE, immunoglobulin E. The immunoglobulin E also is a very small quantity antibody in the serum. It is not found in large amount. If it is found in large amount, then it is an indication that the person is suffering from some kind of allergic reaction or hypersensitivity. Fortunately enough, most of the healthy individuals do not have it in large quantities. Why? I will tell you. The amount that is found in the serum is approximately about 0.3 microgram per ml. If it increases by about 10 times from 0.3 microgram per ml, that itself is an indication of an individual showing allergic or hypersensitive reaction. The major antibodies that are found against the antigens that cause hay fever, asthma, hives, anaphylactic shock, as well as angioedema, urticaria, all these are because of the IgE being present in large quantities. As I had mentioned, IgE, like the IgM, has got 4 constant region domains. Just see here, it is made up of C epsilon 1, epsilon 2, epsilon 3, epsilon 4. 4 constant region domains in the heavy chain. And it has two disulfide bonds, one extending between C epsilon 1, epsilon 1, another extending at the junction between C epsilon 2 and epsilon 3. So two disulfide bonds are there holding the two heavy chains together. So IgE molecule primarily, when it is produced by the B lymphocyte, it immediately seeks a receptor to attach. It is hardly seen in free circulation, therefore it is present in very small quantity, 0.3 microgram per ml, and its molecular weight is approximately about 190,000, and it has a carbohydrate residue attached to the constant region domain, that is C epsilon 2 and epsilon 3, about 12%, and it is found, its half-life is about 2.5 days, 2.5 days. And major physiological role of the IgE molecule is to cause allergic reaction or inflammatory reaction, because it is found attached to the inflammatory cells like mast cells and basophils. Mast cells and basophils have Fc epsilon receptor on their surface. Since they have Fc epsilon receptors, and as I had mentioned earlier, wherever there is blood vessel, there the endothelial lining will be seen, because blood vessels are lined with endothelial lining internally, and wherever endothelial lining is present, there the mast cells will be present lodged within the endothelial lining. So those mast cells are granulocytes and have large number of stored granules in their cytoplasm. And since they have Fc epsilon receptor as shown in the diagram here, when it is attached by the IgE molecule which is secreted against a particular allergic antigen or allergen, two IgE molecules which come and attach simultaneously to the Fc receptors and sit side by side will become the receptors for the antigen to come and bind to their FAB site. Now here you can see in the diagram, two IgE molecules are cross-linked by an allergen or antigen. As soon as this happens, the IgE molecules are capable of causing activation of the mast cells through signal transduction. Why two IgE molecules? The reason being, IgE has a very short cytoplasmic domain. The cytoplasmic domain of the IgE plays a role in signal transduction. Since it is a very short domain in the cytoplasm, single IgE molecule will not be sufficient to trigger the signal transduction. Therefore, two IgE molecules sitting side by side attached to the Fc receptor of the mast cell, and when they are bound by an allergen, which is an antigen, they can trigger the mast cell to undergo activation. The mast cell that undergoes activation leads to degranulation of the granules within its cytoplasm, and those granules have stored histamine, serotonin, prostaglandin, bradykinin. Such pharmacological activators which are found in the granules are going to be released by exocytosis of those granules. As soon as the antigen has established the cross-linking between the two side-by-side sitting IgE molecules, activation of mast cell takes place, and these pharmacological mediators are released from the cytoplasm of mast cells. And that release of pharmacological mediators causes the allergic reaction and inflammatory reaction that is experienced by the person whenever the person comes in contact with the allergen. And whenever hay fever takes place, there will be extreme sneezing and exudation of nasal fluid coming from the nasal region. Or if there is asthma, you know that the constriction of the bronchial passage will take place because of the release of pharmacological mediators, and person finds it difficult to breathe. If the histamine reaction has taken place in the skin surface, the person will find there is severe itching on the skin surface because of the contact of allergen. It may even cause the urticaria, means wheal and flare reaction, and swelling on the skin surface at different regions like islands of swelling. Red patches will be seen, and all these is because of the release of the pharmacological mediators by the mast cells, which is triggered by IgE. Therefore, IgE also is called as reagenic antibody. Reagenic, REA GENIC, reagenic, means it is an antibody which causes the reactions against the allergens. So, the IgE molecule found most of the time attached to the mast cells or basophils. Basophils also, like mast cells, have granules in their cytoplasm, and these granules undergo degranulation and release the same type of pharmacological mediators as I had mentioned earlier for the mast cells. Therefore, one need to be thankful that IgE is not found in large quantities in a person until unless the person is in contact with an allergen or an antigen which causes the allergy. Most of the time it is found in very small quantities. So this is as far as the different classes of the antibodies like IgG, IgM, IgA, IgD and IgE. All these 5 different classes of the antibodies perform different functions. They are found in different regions of the body and they are responsible for tackling the antigen. It is also found that IgE is fortunately enough not capable of crossing the placental barrier. Unlike IgG molecule can easily cross the placental barrier and reach the fetus. Fortunately enough, IgE cannot cross the placental barrier. Therefore, the fetus is saved from any kind of allergic reactions if a mother suffers. Then it is responsible for the alternate pathway of complement to be activated. And it is also a major antibody that is found responsible for attacking the helminth parasites which are found in the intestine. And sometimes when it recognizes the helminth parasites in the intestine of a person, it is capable of binding to the antigens of the helminth parasite and trigger that allergic reaction in the intestinal area, which causes the inflammation, and that is how when there is helminth infection, the person may suffer from the cramp in the intestinal area. The pain or the cramp that is experienced periodically by the person is because of the IgE reaction that is shown by the activation of the mast cells, which are capable of causing the release of those pharmacological mediators in that intestinal area where the helminth infection is seen. So therefore, helminths are majorly tackled by the IgE, as well as at the same time it is also tackled by IgG. So you know now the allergens or the antigens that cause allergy majorly responsible for triggering IgE production, and it is the major cause of allergic reaction. So let us see now another aspect of the antibodies. The antibodies were classified not only based on the type of the heavy chain domains like gamma, mu, alpha, delta, and epsilon. The antibodies also can be classified based on the antigenic determinants. The antigenic determinants that are present on the antibody, in the sense the amino acid sequences that are there in the antibody, are responsible for production of antibody against antibody when an antibody is injected into another animal. Let us say, suppose if you inject the human antibodies into rabbit or sheep, rat or mice, they recognize this antibody as a foreign antigen and they produce antibody against the antibody, and those antibodies will recognize certain regions of the antibodies as antigenic epitopes, and the antibodies that are produced against the antibodies will bind to these antigenic epitopes. Based on this, the presence of antigenic determinants on the antibodies, the 5 different classes of the antibodies are classified into 3 major categories. That is, depending on antigenic determinant. As mentioned in the diagram, one is isotypic determinant, second is allotypic determinant, third will be idiotypic determinant. And each of these determinants are present in different regions of the 5 different classes of the antibody. And based on that, one can also classify the antibodies as isotypic, allotypic, and idiotypic. Now first I will take up the isotypic determinants. Now here in the diagram, on the extreme left hand side, the simple antibody molecule in the Y-shaped form is shown, a simple typical antibody molecule. And on the right hand side as well as in the middle, 2 different antibodies have been shown of the mouse origin. One is IgG1 and another is IgM. And IgG1 is a subclass of IgG. Now you take into consideration when these 2 antibodies, let's say the IgG as well as IgM, when introduced into another animal, they are of mouse origin. When they introduced into another animal, that is rabbit, they triggered the production of the antibody in the rabbit, and the rabbit antibody is capable of binding to the specific sites on these 2 antibodies, and those sites have been represented by different colored regions. As far as the constant region domain of the IgG is concerned, you can see with the different shapes, the determinants have been shown, as well as the constant region domain of the light chain also been shown with a different shape epitope. So the antigenic determinants of IgG, you compare it with antigenic determinant of the IgM. They are of different types. Therefore, they have been shown with different shapes. They don't show actual shapes in the real situation, but this is for your understanding purpose. You can see here in the case of IgG, the shape of the antigenic determinant in the CH1 domain of the heavy chain is shown by an arrow, whereas here it is shown like a dumbbell in the case of IgM. That means these two regions have different amino acid sequences, and these two antibodies differ from one another because of the difference in amino acid sequences. And these amino acid sequences which are different in the constant region domain of both light chain and the heavy chain act as epitopes for the production of the antibodies, and each of these epitopes produce a different antibody, and they are specifically capable of binding to these epitopes individually. So the different classes of the antibodies, that is IgG, IgM, IgA, IgD and IgE, are classified under isotypic determinants because each of these not only differ in their heavy chain constant region, they also differ by having different amino acid sequences. Therefore, they trigger the production of antibodies which are specific to these antigenic determinants, and the antibody that is produced against the IgG molecule against these antigenic determinants will not be able to react with the antigenic determinants of the IgM. In the same manner, vice versa. That is, antibodies produced against the antigenic determinant of IgM will not be able to react with the antigenic determinants of the IgG. So therefore, they are capable of individually recognizing each of the classes of the antibodies and differentiate them based on the antigenic determinants. So this is how, using the antibody molecule and triggering the antibody production against it, one can find out how they differ from one another and why they are classified into different classes. So isotypic determinants are responsible for the classification of the antibodies into 5 different classes. As far as allotypic determinants and idiotypic determinants are concerned, we will see it in the next class. Thank you very much.